Hi everyone, welcome to today's webinar. I'm Charlotte, I'm the COO at Sano Genetics. At Sano, we combine genetic testing, recruitment, and long-term engagement into a single platform to accelerate enrollment and simplify operations for precision medicine studies. I'm delighted to be here with all of you as we dive into a really important topic, rare disease research and drug development. And I'm thrilled to introduce our special guest today, Harriet Holme, Chair and founder of PCDresearch.org. Harriet, you're a clinician, but you're also an exceptional leader in the field of rare disease, which is a powerful intersection of expertise. So thank you so much for joining us today.
Thank you. It's great to be here.
Harriet, before we dive in, I'll just outline what we'll talk about today. So we're going to have a look at your background and what drew you to this field. And we'll look at recent breakthrough in rare disease research and the challenges as well in developing therapies. We know that it's particularly difficult for rare disease, as well as solutions for improving the drug development process. And then we'll close with some key takeaway and opportunities for collaboration. So let's dive in. My first question, Harriet, is can you start by telling us a little bit about your background and most importantly, what motivated you to specialize in rare disease research?
So a little bit about me. So actually, I read medicine at Cambridge, and then I was an NIHR academic clinical fellow. I worked in the NHS in paediatrics, and I specialised in paediatric oncology. I really, there's obviously a huge unmet need in paediatric oncology, but thankfully not many children have cancer. But I really enjoyed not only helping and treating those patients, but also understanding, sort of the precision and the research behind oncology. And so it was then that I went on and did my PhD. So I did a PhD in synthetic lethality of osteosarcoma. I did that at the ICR at UCL and in collaboration with the Sanger. Alan Ashworth was my PhD supervisor, and I'm still very much in contact with him. He now heads up the oncology department at the UCSF. And I really enjoyed doing lab work and understanding the biology. And it was sort of then that my son was born, and he has a rare disease, and that had then changed my life course. So I didn't end up as the professor of pediatric oncology that I thought I was going to be. And I moved into drug development. So I work at Wetherden. Wetherden was set up a few years ago, really with that sort of the idea to test whether trials fail because of biology or whether there were other reasons. And industry average for success for early phase clinical trial is 29%. At Wetherden, where I work, it's 65%. And I think that really demonstrates that actually biology is not the main reason that clinical trials fail. So at Wetherden, I work across all assets and indications. I help with biotech and early mid-cap pharma, look at lifestyle management, indication selection, scientific rationale, understanding commercial placement, writing their TPP, their target patient population, and their synopsis. The areas that I enjoy are in oncology, advanced therapeutics, respiratory medicine, and rare disease. Those are my sort of particular focus, but I work across a whole field. So it was really, though, during that understanding, sort of around the sort of same time, I guess, I set up PCD Research, though the charity that I chair and founded, because there were no other charities looking at primary ciliary dyskinesia and funding research with that focus on trying to get a drug to the clinic. So primary ciliary dyskinesia is where the sort of hair-like structures that are all over your body, but predominantly in your lungs and sinuses, they should sweep the mucus and the debris out of the lungs, but they don't work. So there's a problem with the motility, and that causes problems just like cystic fibrosis. So cystic fibrosis, the mucus is very sticky and doesn't move out, but it's that same mucus, it causes very similar problems. So it causes pneumonia and a relentless and irreversible lung function decline. And astonishingly, even though it's got a very similar market size to cystic fibrosis, so there are about 150,000 people estimated worldwide to have it, and there's been multi-billion, multi-modality asset investment in cystic fibrosis. There was nothing in primary ciliary dyskinesia. And so I thought, so there are no evidence-based treatments. There are no FDA, MHRA, no approved treatments. And And so really it's just palliative options, because there's no way of stopping or halting that lung function decline. And I really thought, something had to be done. And so I set up PCD research. Within a year, we were a full member of the AMRC. We've raised a couple of 100,000. We have partnered with Nucleic Acid Therapeutic Accelerator to jointly fund a 2-year postdoc to look to see if we can functionally restore cilia, first in cell culture and alley culture. And then we have a collaboration as well with Harwell to then put that into a mouse. And there've been technical challenges with PCD mice, but trying to sort of overcome those and really sort of understand as a proof of concept how we can start to make functional restoration in PCD and work towards approved therapies and disease-modifying treatments in the clinic. So that's what's driven me, I guess. That need to do better, I think, across rare disease. My background being a pediatric oncologist, if I look across at something like acute lymphoblastic leukemia, which a few decades ago was universally fatal, and through those well-designed randomized clinical trials, we've got this sort of event-free survival of more than 90% at 5 years. Acute ALL is 4 times rarer than PCD, and yet we've only had a couple of RCTs in PCD, and we've made almost no progress, in my opinion. So I think it's really understanding the value of clinical trials, the value of a well-defined genetic cohort of patients, understanding their phenotype, and understanding how you really shift the needle on treatments. So through all of that, I raised money, thought if we just did some research, then we could start making progress. But it was actually understanding here, research isn't one, actually I could fund millions of pounds worth of research. I'm not entirely sure that's the block. I think it's an important part of the parcel, but actually the rare disease landscape is really challenging, and it's challenging for investors and challenging for industry. And that's one of the reasons why it's so challenging for patients, that just 5% have got approved treatments.
That's really fascinating to hear everything put in place. Why do you think that is the case? Is it because, to your point, there is less research, people are less aware of these conditions because they're rare, or is it more economics to some extent? Like, because they are so rare, there aren't many industries or pharma companies willing to run clinical trial in these conditions. What's the main blocker?
Well, actually, so I've become really interested in putting all of rare together. And I think this can't move the needle on just focusing on one disease, but actually focusing across rare disease. So across rare disease, rare isn't rare. It's 4 million, it affects 4 million people in the UK. So 4 million people are living with a rare disease. And that's 1 in 17 people. And then if you imagine each one's got a carer and impact on the family, it affects 1 in 7 people in the UK. So this is a really big problem for an awful lot of people. But I think what's not there is the economics. And I think that there's no value, there's sort of a very limited commercial value proposition within it. So clinical trials, so through my work as a drug development clinician, I understand that the drug that you come up with, the clinical development pathway is determined by time and money, how long it takes and how much it costs. And so if it takes a long time and costs a lot of money, you're going to have a drug that's very expensive. And actually, if you've got a very small market size, it's just not commercially attractive. And I think part of the problem with rare disease is it's got a lot more challenges than, say, a common disease where you could overcome some of those problems. So there's a big problem, I think, with access, with getting a diagnosis. So we know that already it takes 4.7 years on average to get a diagnosis if you've got a rare disease, even though 80% of people have already got identified genetic origin. So those people should be identifiable from conception, but we're not identifying them. So by the time they're identified, they've already got end organ damage. And it's huge and costly, not just in terms of the mental health burden for the patients, but also for the Department of Health. It's been estimated it's cost the Department of Health over 3 billion over the last 10 years in that diagnostic odyssey. So that there's multiple failed hospital appointments, the trauma for the patients, the trauma for the parents, the carers, the families, the loss of productivity, the loss of impact on education. And 30% of children with a rare disease die before they're 5. Some will die anyway because sadly there aren't treatments. But I think we shouldn't be in a position where children are potentially dying before they've got a diagnosis. And actually, when that diagnosis is, possible from conception. So we've also got a sort of two-tier system where some rare diseases, a tiny percentage of them are on the newborn blood spot test, and the rest of them aren't. So actually, if you've got cystic fibrosis, you're identified at birth, you can start chest physio and input, and actually it's then much easier to do drug development if you've identified the population. If you haven't identified the population, so in PCD, there are estimated to be 9,000 patients living with PCD in the UK. Only about 1,100 have been identified. So we're missing about 8,000 patients. So it's really hard to run clinical trials when you haven't identified and found the patient population. If you can identify them, put them on a registry or they're accessible, then you can run clinical trials. So I think it's a sort of chicken and egg. We don't identify rare disease because there aren't any treatments, but there are never going to be treatments if you can't identify them. And then problems with it's sort of there's the sort of regulation and manufacturing angle. So I think that an awful lot of these rare diseases are really amenable by genetic therapies. Obviously, there's sort of drug, small molecule, drug repurposing. But if you think about a sort of durable one-off, think about CRISPR, or even just sort of that sort of longer term treatment, something like mRNA, you could functionally restore and replace the faulty genetic product, that protein, and so you could do that in patients with a rare disease, but part of the problem is that sort of genetic heterogeneity. So if I look at and use PCD as an example, cystic fibrosis, it's homogeneous. It's just one gene, delta FL8. With PCD, there are 1,000 proteins in the cilia. Only 56 disease-causing genes have been identified. We're not doing whole genome sequencing. We're just doing a limited panel. So that makes it really difficult to identify. Almost certainly there are going to be other genes. And so how do you then start addressing diseases, particularly like PCD, where it's structurally complicated? It's almost certainly not going to be, you're not going to get functional restoration of a cilia through a drug, because of the sort of complex nature of the movement. So you're going to need those genetic therapies. If you think then of the regulator, say you've got an LNP mRNA platform and you could address 56 genes across PCD, for each and every single one of those genes, you would have to do a CTA application, you'd have to do all of the tox and all of that, you'd have to go through everything all over again for each individual gene. And that just makes it, for a tiny population, because if you've got PCD and then you're splitting it effectively into multiple different ultra rare diseases with a different genetic origin, then it just makes it commercially unattractive. Or actually, part through my work at Weatherden as well, we're having these conversations about how actually we could align the regulation and the manufacturing so that, for example, you could use gene 1, use it as proof of concept, and then amend your CTA application to add in prior knowledge. So actually then make it a lot more streamlined and to make those genetically heterogeneous diseases a lot more addressable, and have the same with the manufacturing, that you shouldn't have to start all over again when you're just doing small changes. You're changing the vehicles, so
So to some extent, there are some economies of scale that need to happen for this to be viable. What role do you see, NICE in the UK or the Orphan Drug Act in the US playing a role into these treatments potentially being developed?
I think there's obviously some help from the different sort of pathways that have been set up to facilitate orphan drug development, and they have been helpful. I think though that so if I use cancer as an example, it's really demonstrated how that sort of industry pressure has helped shape things that maybe would have been difficult to regulate. So autologous CAR T, cancer vaccines, these should have been quite difficult, but actually, that hasn't prevented innovation, probably because there's more investment and pressure to do so. I think as a rare disease community, although it's engaged, it's quite fragmented, and so it needs to come together and actually say, well, this is what we need now. And so while other things have been helpful, I think that this is still a major block for investment, and I think that would really, really help. I think other things along the pathway needed. So speeding up, making clinical trials more efficient so that you can get the patients in much quicker and you're not having the same challenges trying to find them. I think
And I think to your point on this prenatal diagnosis is quite key for rare disease. So I imagine you looking with with interest at the 200 genetic newborn screening being launched in the UK, and whether we could replicate that on a larger scale and in more countries would be really interesting, I think, in terms of breakthrough for rare disease clinical trials.
yeah, absolutely. So the generation study is going to sequence 100,000 babies. And obviously, that's really small for individual diseases, but if they come together through a consortium approach to something like through ICON, where different groups are doing it all over the world, there's power then through combining that data. And I think we really need I think we really need to move into the 21st century and start really harnessing this. And actually although it can feel sort of scary for people having, a well baby in front of them and doing their genetics potentially, I think that, ask anyone with a child with a rare disease, and, that having a diagnosis would have made a huge difference, and not having, all of what we talked about before. And so I think it's, weighing up that societal cost versus weighing up that huge benefit for the individual. And I think actually it's only through that early diagnosis, timely diagnosis and before end organ damage, that having that defined population will make drug development a lot easier. We know that even for just small molecule development, understanding the genetic pathways, understanding the genetics increases the success of drug development by 2 to 3 times. So this is going to be really key to development.
Yeah, I was going to say genetic studies, but also patient-derived models have been game changers in research. So these tools can accelerate the development of therapies for rare disease, hopefully. I'd like to shift gear a little bit and talk about breakthrough that we've seen in the past years. I mean, you mentioned newborn screening program. We obviously have like the cost of genetic sequencing reducing over time, which is making it more accessible to do on a large scale and earlier. What other exciting advancement have you seen recently that you're particularly excited about and you think might pave the way for a different future for rare disease patients?
I think it's only 11 years ago that I was doing CRISPR in the lab, and the speed at which that's really coming towards the clinic. Obviously this had the first approval, but I think that over the next few years we're going to see that really accelerate, and I think that's really exciting to potentially be able to dose to treatment, to dose to effect, have that one-off treatment that shouldn't be immunogenic, and actually really address potentially, I'm cautious about using the word cure, but have that sort of durable effect. I think for that genetic rare disease, it's an inherited rare disease. I think that's the most exciting thing for me. But I think in order to do that, the landscape has to be ready. And so actually the scientific progress is there really. That's not sort of rate determining step. It's actually all of the rest of the bits to make that CRISPR commercially viable when we get there at scale. And I think it's going to be that having these things at scale will be the exciting part.
Yeah, so that's kind of my next question. Like looking ahead, what practical solutions do you see in overcoming these barriers? I mean, I know you and I spoke about basket trials when we last met. How do you see us overcoming some of these issues, particularly in the UK, where we have some barriers, in terms of funding, regulatory processes, and so on?
So I actually think that the UK is uniquely positioned for success in this arena. And I think that's because we've got the largest phenotypic data set in the world in the NHS. We've got a single provider of sequencing, and we're not having to rely on individual companies in quite the same way. So you could have it much more aligned with that NGRL, the National Genomics Reference Lab. And I think then if you integrate that data, you integrate it with biobank data as well, you have a really powerful data set to really help patients. And I think that is key to why the UK is uniquely positioned. And I think then, we've got tech now like LLMs, large language models. We should be able to move past things like ICD-10 codes, which really don't account for most of rare disease, and actually then be able to pull patients out, not just once they're diagnosed with an ICD-10 code, but you could actually then pull them out before they're diagnosed. So I think it's two things: that top-down approach to diagnostic uncertainty. We should be much quicker at getting the genetics done. I still think that we need to be embedding whole genome sequencing a lot more in the NHS. I don't feel that we're there. And also, I think, at birth, we should be doing, I hope that we'll build off the back of the generation study and we'll really start to see changes within that, as soon as possible, really. I think, the more we can diagnose babies before they have end organ damage, better. And I think then combine that with regulatory and manufacturing changes and streamlining clinical trials so that you can pull patients out much more effectively, so without the sort of barrier of trying to find patients. You would then not need a registry if you had data integration and that LLM, because you could find patients much more easily. We wouldn't be reliant on clinicians trying to find time within busy clinics to enter data. And also, there's problems with the inclusivity of getting all of the rare diseases onto one registry. All of that has been a big problem. Actually, we could run this all a lot more efficiently. And I come back to, that sort of drug development question. This is all time and money. And actually, you reduce the time and you reduce the money, then you make drug development actually happen. And I think that's the key here. If you align all these different bits of the landscape, then it makes a value proposition. And I think everyone's a winner then, because actually you get massive capital investment, you get growth of UK PLC, and you get treatments for patients with rare disease, which is, the primary driver here.
And it's kind of the hard part, isn't it? Because everyone is working in silos. You have government making policy to try to unblock some things, and then you have pharma companies or biotechs that are running clinical trials, but within their own dedicated pipeline, with a sort of ROI calculation on top of this. And then you have patient advocacy groups and others, that are sort of sitting a little bit on the side of all of this landscape. So how in your line of work have you bridge that gap to some extent? How do you make all of these groups talk to each other so that they can concentrate efforts, and sort of row in the same direction?
I think that's really challenging, and it's certainly something that, hasn't been easy. And also trying to align everyone to the same direction is, it can be challenging. I actually ran our, as sort of with both hats on, from my Weatherden and also PCD research hat. We ran our industry accelerator day on the 1st of October at the Francis Crick Institute, where we brought 120 people together. They were sort of 40% from industry. We had 10% VC, so as investors. We had NICE, Department of Health. We had policy makers. We had some IP lawyers. We had some patient advocates. We had clinician scientists, to try to bring everyone together, to actually bring everyone, first of all, onto the same page, to actually, sharing some of those problems and sharing the solutions about how we could address them.
Yeah, no, that makes sense. And I guess, I kind of like a little bit of an optimistic note in all of this, because it seems hard work, and sometimes you just feel like you're never going to manage to get all of these players to just sort of, again, row in the same direction. But we have had success stories, haven't we? You mentioned ALL earlier. We are certainly seeing conditions where just a decade ago, would not have a treatment, and now there are options.
Well, I
Yeah.
think a case in point of that is cystic fibrosis, right? Because actually, historically, it was considered too difficult to treat. Had this small group of patients, or considered to be small. Actually, it's quite a common rare disease, but a patient population that was very sick and was considered, very challenging, very difficult. And actually, now look with CFTR modulators, we've got a population where 85 to 90% of people have got disease modification, which is, it's an incredible achievement. Yeah. I think though that you can see why it's worked so well in CF. They're on the newborn blood spot test, so they're identified at birth. There's a really well defined patient population, an excellent registry, so it's easy to run clinical trials. And there's been, decades of multi-billion pound multi-asset investment. And still, even now, that's still going on despite 85-95% being addressable to CFTR modulators. There's still
Yeah.
billions and multiple assets, that sort of race to get to that 10% really. And you can see how some rare diseases have actually overcome some of those challenges on a disease by disease level. And then others, so I put PCD side by side, we don't have that at all. And it's the same, similar size population. So it's not just about the size of population, it's about those other factors as well.
Yeah.
And I think that's key.
Yeah, I feel like we are that much closer with monogenic disorders where we're starting to get the technology to potentially address them. And mind you, we haven't yet, right? There are still many out there that, haven't, where a treatment option isn't available. But I feel like we have the convergence of the genetic understanding of the condition and the tools to potentially address it. And my hope is that it's a low-hanging fruit in the next decade for those to potentially have treatments that weren't available before. But it's a lot more complex when it comes to polygenic conditions or to conditions that have more complex genetic needs. And I'd like to think that eventually we will crack it. But to your point, it's about the right resource and the time dedicated to it. And we have just so many tools becoming available at once and so many breakthrough in technology, and just where we apply them to disease and what we prioritize at the moment is just, it's all over the place.
Yeah, so I do actually think that ultimately this is entirely possible. I think I just think it's going to be, sort of buy-in to shape that landscape is going to be the key part, actually getting, sort of everyone aligned to that, end-to-end solution to facilitate that innovation. And I think that's the bit that's really important. Actually, I think innovation, I don't think we'll have a problem with innovation. And I think a lot of the tools are there already and are very nearly ready. I think it's that, that ecosystem to facilitate innovation that's key.
Yeah, I mean, and I feel like in that sense, I have a similar background to you. I also left my research world with the conviction that we weren't short of scientific insights, and that there were plenty of incredible scientists out there, that were generating papers and insights at an unprecedented rate. But ultimately, the real blocker is in then taking those insights and turning them into treatment for people. And, the clinical trial process and the economics of that and the speed of that and the complexity of that has a lot to answer for, and like a big role to play in that.
Yeah, absolutely. And I just think, when I started back doing my sort of ACF, 15 years ago, I was doing Sanger sequencing by hand because it was, too expensive to do whole exome sequencing, and, it was thousands. And, look how, affordable it is now. Actually, we've got amazing tools and we've got the resources. It's just about, really judiciously using them. And I think, actually bringing some of these diagnostic techniques more into the future, more into today, and more more on just I just really think that we need to have that greater genetic molecular characterization of these conditions would be really helpful for everyone. And I think really bringing that up to speed is key here.
Yeah. Yeah, I mean, I think it's really interesting as well. We have initiatives. I mean, you studied at the Sanger as well, right? The human cell atlas, the fetal cell atlas are just like really interesting initiatives as well that I think are starting to become more public, and that are really playing a key role in us understanding not just the genetics, but the cellular status and just the different moving parts that underpin that biology. So.
Yeah, absolutely. So I did my PhD at the ICR, but I collaborated with the Sanger to do sequencing for my models. But I think genetics have been so important in cancer. Look at the sort of understanding of synthetic lethality, those targeted treatments. And actually, if we didn't have that molecular characterization and understanding, we'd still be using, what we wouldn't have those targeted therapies in the same way that have been really game changing for cancer. And I think we now need to bring up the rest of, particularly rare disease up to that level. If molecular characterization is a standard for cancer, not just at diagnosis, but also, at sort of malignant, that metastatic phase, or, understanding that clonal evolution within the tumor, that actually, that should be priority for rare disease patients. A molecular characterization, so genetic diagnosis, should be really a foundation, I think. And I just don't see that happening at the moment.
Well, you kind of answered my last question, which was around what do you see as the future of precision medicine in addressing those needs? And we've talked quite a bit about the barriers to getting there. So that's my sort of parting thoughts here is, for everyone listening, is how do we break down these barriers and how do we collaborate, with the different groups that are pushing for a change in that, in that respect, to really get the needle moving.
But I think by aligning that infrastructure and those pathways that we've talked about, we've got that, a real opportunity to create that economies of scale across whole of rare disease. And you can then use that ecosystem for drug development and make it economically viable. And it would really position, I think, us as a world-leading innovator, and it would attract that growth, investment. And I think actually we then have this opportunity not just to address that significant unmet need and social justice and lives not lived in rare disease, but use that as a sort of scalable model, as a precursor for more personalized medicine across the whole of health. Actually, it's sort of a new model, and I think that's really exciting.
Yeah, I do too. Harriet, thank you so much for a fascinating and insightful discussion. It's clear that the work you do is really, like thriving to make a difference in the field of rare disease, particularly in PCD, and I'm really excited to see what comes next. To everyone joining us today, I hope you found the conversation as insightful as I have. We'd love to stay in touch, so you can learn more at sanogenetics.com, pcdresearch.org, and weatherden.co.uk. Harriet, is there anything else you'd like to highlight before we wrap up?
No, thank you. And thank you to all of the people, really, I guess, that, I talk to regularly about how to, and what sort of partner and collaborate, and, really want to really have that, bold and ambitious change for rare disease, and help those millions of patients.
And there are quite a few of us out there, so get in touch. Thanks, Harriet.
Thanks very much.